15-minute or less lecture series, Anatomy and Physiology, Chapter 3, Cells, Part 2. We're now moving on to the cytoplasm, everything between the nucleus and the cell membrane. Within the cytoplasm is the cytosol. This is the aqueous solution, the watery solution inside the cell, which is actually kind of gel-like. It also includes the cytoskeleton, long protein rods and tubes that give the cell strength, gives it structure, and helps to organize various materials. And then also the organelles, structures that have specialized functions inside the cell. One organelle is the ribosomes. There are thousands, tens of thousands, who knows, lots of ribosomes inside of the cell. They're small organelles. They can be found free floating in the cytosol, in the fluid, or they can be bound to the endoplasmic reticulum. Ribosomes do not have a membrane. They are just proteins and RNA, ribosomal RNA, that is bound up in a functional cluster. And the function of this mega enzyme is protein synthesis, engaged in a process called translation to make specific proteins. Ribosome, the rough endoplasmic ER. The rough endoplasmic ER is flattened interconnected membrane sacs that are located throughout the cytoplasm. They are attached to ribosomes. This is what makes them rough or bumpy looking. And they're involved in protein synthesis and processing. The ribosomes make the proteins. The rough ER will then help the proteins to fold, will help to modify the proteins, and then send them on to the endoplasmic, the Golgi apparatus. Then we have the smooth endoplasmic reticulum. This is a little bit more cylindrical in shape, but it is still interconnected membrane sacs. Smooth because there are no ribosomes here. Instead, it involves in lipid metabolism, in the synthesis of some lipids, in fat absorption, and also the breakdown of some drugs. Then there's the structure called the Golgi apparatus. It is a stack of flattened sacs made of phospholipids. It receives proteins from the rough endoplasmic reticulum and then modifies them, packages them, and transports them on. Often the thing it adds is the carbohydrates, the glyco part of glycoproteins. And when the materials are sent out of the cell, it is referred to as exocytosis. So here is the rough endoplasmic reticulum sending vesicles filled with proteins to the Golgi apparatus, which then sends the proteins to the different levels, processing them, modifying them, and then sending out vesicles either to fuse with the cell membrane, to incorporate proteins into the cell membrane, or to fuse with the cell membrane and release proteins to be secreted, or sometimes to form organelles such as lysosomes. Mitochondria. Mitochondria is a double membrane structure. It has two phospholipid membranes. It is where ATP is produced. So cellular energy is made in the mitochondria. And this special structure is able to divide and separate on its own. Lysosomes are modified vesicles. These modified vesicles are filled with digestive enzymes. What lysosomes do is, for instance, when materials are brought in via phagocytosis, the lysosome will fuse with that vesicle and then break down the material or pathogens or whatever that's inside that vesicle. Lysosomes can also break down and recycle old organelles and can be involved in the destruction of some drugs, toxins, and cholesterol. Peroxisomes. Peroxisomes is another membrane sac filled with enzymes. These enzymes have functions such as breaking down lipids into smaller structures. It also neutralizes hydrogen peroxide, that's where the organelle gets its name, peroxisomes, peroxide. It turns out hydrogen peroxide is a metabolic product that we have our cells make, but is toxic, so it needs to be neutralized. And peroxisomes are also involved in detoxification of alcohol. Cytoskeleton is a structural component to the cells found in the cytoplasm. It is made up of long protein rods and tubes that do things such as give the cell its shape or reshape it to allow the cell to move. It can be an anchorage site for organelles in the cytoplasm, a roadway for vesicles or organelles to move along, and also is important in moving chromosomes during cell division. There are two main types of protein structures. There's the microfilaments. These are rods made of the actin protein, so you have thousands, millions of actin proteins all forming long rods. They're often associated with the cell membrane and are involved in cell shape and movement. There are also microtubules, long chains of proteins that form a tube-like structure that is found throughout the cytoplasm and is often a roadway for vesicles or holds organelles in certain places, and also can be involved in moving chromosomes during cell division. Centrosome. Centrosome is an organelle that has no membrane, and it is where microtubules are produced. In the centrosome are two special cylindrical structures called centrioles that are the most active portion of the centrosome. And again, centrosomes do many things, including having a large role in helping to separate chromosomes during cell division. On the external portions of the cell, there are a few cells out there that possess either cilia or flagellum. These are motile extensions from the cell membrane. The only cell that has a flagellum is sperm. They are the only ones that have a flagellum, and it is there to move the entire cell. Cilia, on the other hand, come in many, many hundreds or thousands or even more on the apical surface of a cell, on its free surface. And the cilia's job is to move materials outside of the cell. This could be mucus that is lining a structure such as the trachea, or it could be other things, fluids outside of the cell that need to be moved. Moving on to the nucleus. Nucleus is the largest organelle that houses the genetic code, the DNA. The nucleus has a nuclear envelope, which has a double membrane layer, so two phospholipid bilayers make up the nuclear envelope. There are also very large pores that are there to allow large materials to pass into or out of the nucleus. The fluid in the nucleus is called nucleoplasm. The darkened area inside the nucleus is considered an organelle, and it is called the nucleolus. Nucleolus is where ribosomes are produced. Ribosomes are produced in the nucleolus, leave the nucleus, and then produce proteins of their own. Also found in the nucleus is the DNA. However, the DNA is not naked floating around. It is bound up with proteins. So DNA bound up with proteins that are in a relaxed state are called chromatin. So chromatin is found scattered all throughout the nucleus. So here we can see naked double helix of DNA. It gets bound up with the proteins to form the chromatin found in the nucleus. And during cell division, this chromatin can condense even further to form chromosomes. Chromosomes that look kind of like an X and often have an area where they're connected called the centromere. All right, life of a cell. Turns out many cells have a cell cycle. And in the cell cycle, they have the interphase, where they engage in most of their functions, so the normal cellular functions the cell engages in. And then also another phase, a metaphase, where they can engage in division. So a new cell starts off in the G1 phase of interphase. And some cells then leave the cell cycle to do whatever it is they need to do and never divide again. However, other cells that are going to divide will then grow some more and move into the S phase. In the S phase, they will then duplicate their DNA. Make two copies of the DNA so that when they divide to form two new cells, each cell will have a complete copy of the DNA. Then the cell enters the G2 phase of interphase. Here the cell grows even more, producing more cytosol, more organelles, so that the two new cells that are formed will have enough materials in their cytoplasm to survive. The next phase is, of course, the cell division or mitotic phase. And during the cell division, there are two main processes: mitosis, which is division of the nucleus, aka division of the DNA, and cytokinesis, which is division of the cytoplasm, the entire cell as a whole, becoming two. So we have interphase. One parent in interphase eventually reaches the G2 phase of interphase. Then we'll enter mitosis, going through phase, metaphase, anaphase, telophase, where cytokinesis also occurs to give us two identical sister cells. So, interphase, again, this is the growth phase. This is when all the cellular structures mass produce. You end up with two centrosomes, with two pairs of centrioles, two copies of the DNA. You have a nucleus present. Everything is as expected. Then the cell is going to divide will enter prophase. During prophase, the nuclear envelope will break down and the nucleus will start to disappear. The chromatin will condense down to form the chromosomes. Each chromosome has two sides. Each side is a chromatid, so there are two sister chromatids connected by the centromere. The nucleolus will disappear, the endoplasmic reticulum will disappear, and the centrosomes or centrioles will start to migrate to opposite sides of the cells and start producing spindle fibers, specialized microtubule structures. Cell will then enter metaphase. Here, the chromosomes all line up down the equatorial plate or midline of the cell. The centrioles of the centrosome will then stretch out their spindle fibers that are building and building and building, and then connect to the centromeres of the chromosomes. Then during anaphase, this mitotic spindle will then shorten and the spindle fibers will start to drag the chromatids toward separate ends. Once the sister chromosomes are broken into separate sister chromatids, then they are officially chromosomes again, and this way each side of the cell will hold a copy of the cell's DNA. Also during this phase, you'll start to see a cleavage furrow in the cell membrane as cytokinesis begins. Then there's telophase. During telophase, the chromosomes arrive at the opposite ends. The nucleus begins to reform. The chromosomes start to relax and return to chromatin state. All the structures that went away start to redevelop. And cytokinesis will complete, giving us two new sister cells. So here's the cell cycle. Interphase, eventually leading to prophase. Chromatosomes form. Metaphase, they line up. Anaphase, they get ripped apart. Telophase, two new cells forming with two new nucleuses, and it all can go over and over again. Apoptosis is programmed cell death. Sometimes we need cells in our body to die, and it is a good thing. For instance, we all started off as a single fertilized egg that is basically a super stem cell, and it divided and divided to form balls of cells that became specialized stem cells that continued to divide and divide until we got all the different specialized cells of our body. Each of the cells housing same amount of DNA, identical DNA, but only using some of the genes in the DNA that is important for that particular kind of cell. And when we were an embryo, we had webbing between our fingers and toes. And as we further developed, apoptosis caused the cells of the webbing to break down. And that is it for Chapter 3.